Okay, well, thank you very much to the organizers for giving me the opportunity to share our research on microscopic tweezers, which are a new avenue for the treatment of neurodegenerative diseases. First, I'm going to start by giving a brief introduction on what protein aggregation is, on the concept of protein aggregation. And really, this is just a sophisticated word for clumping. In our brain, we have cells. Normally, everyone's familiar with the term of a neuron that allow us to move, think, etc., coordinate many different tasks. Now, these cells require very tiny biomolecules termed proteins, and these are represented here as this little yellow squiggle. Now, in physiological or normal conditions, these proteins go about their function perfectly normally. But then when disease sets on, in neurodegenerative diseases especially, these tend to aggregate. They tend to clump with each other and build clusters, low-grade clusters at the beginning, termed oligomers, and then these can even go on to form further elongated forms like fibrils, and these then build lesions. And you might be familiar with some of the proteins that undergo aggregation in many different neurodegenerative diseases, such as beta amyloid and tau in the case of Alzheimer's disease, or alpha-synuclein for Parkinson's disease, or Huntington in the case of Huntington's. Now, these forms of aggregation are found in the lesions that are allowing us to diagnose people with these diseases. If you think about amyloid plaque, it's full of beta amyloid. If you think about tau tangles, it's full of, or neurofibrillary tangles, it's full of the protein called tau. But why do we care about aggregation? Why is it a problem in the cell? Now, it is a problem because if we think about the neuron depicted in here as this big blue cell, and we have to imagine this process shooting off for meters and meters until it communicates via this terminal with another cell. So this is only here for the purpose of the cartoon, but it would be very far away and communicating with a different cell. If we think about these, they have a lot of maintenance to keep up with their good work. They need to continuously be producing new proteins and degrading old proteins. So production of new proteins normally happens in the endoplasmic reticulum, or ER, and this is impaired by aggregates, again depicted here as these yellow clusters. Now, the clumps don't allow for the new synthesis of proteins. Therefore, the proteins that we get are susceptible to the clumping and are not as functional as a new protein would be. If we think about where they get their energy, the powerhouse of the cell, that's the mitochondria. These organelles are also disrupted by aggregates. They're disrupted by these clumps. And so the cell is not able to charge their batteries. They don't have the energy they need to carry out their function. If we think about the electrical impulses and the communication with other cells at the terminal of the neuron, this happens through a very delicate balance of very, very highly well-coordinated proteins and vesicles, and this is really disrupted by the presence of aggregates. Therefore, the communication is not as efficient. And finally, if we think about waste management within the cell, if we think about how these cells get rid of the old proteins, even some of the clumps of the bins, if you will, which are autophagy and lysosomes and lysosomal flux and proteasomal degradation, these are blocked by the clumps. So very much in the way like when a bin gets full and doesn't get emptied, it no longer is quite useful. So what this ends up doing globally is disrupting the whole cell. And at the beginning, it's a disruption, so it struggles by, but it kind of ends up being a bit too much, and ultimately it can lead to cell death. Now, it's not only intracellular effects that we're interested in many of neurodegenerative diseases. Aggregates can also affect the extracellular, extraneuronal milieu. And there are other cells in the brain that are highly important, microglial cells. They are the vacuum cleaners of the brain. They make sure everything is tidy and everything is spotless and great. for the neurons to actually communicate with each other. And what happens when these vacuum cleaners get saturated with aggregates is they're no longer very happy. They stop removing the clumps efficiently and stop tidying the place, which makes the neurons really unhappy as well. And not only that, if you think about a vacuum cleaner that has been left on for too long and is overheated, it can even burn down the house. So they start even being detrimental for the functioning of the brain. Now, what is a microscopic or molecular tweezer? A microscopic or molecular tweezer is just that, a molecule that looks like a tweezer. And you'll have to use your imaginations here, because often scientists are a bit like that. We kind of see structures and molecules that may not be very easy to see, but this is the molecular tweezer that I work with, CLR01, and the skeleton is depicted here of the molecule in black. If you use your imagination, you can kind of see the shape of a tweezer there, which I'd like to cartoon into this little shape. The important thing about this tweezer is how it works. So again, if we go back to aggregation and our clumpy proteins, if you think about the yellow squiggles as a ball, we can see that in normal circumstances, these proteins, again, work in the same way. In disease, they aggregate, they build these clumps. The way the molecular tweezer would act on this would be to actually bind to certain parts of these proteins and therefore impair the aggregation. It won't allow the proteins to clump with one another, and so prevent the clumps and prevent the detrimental effects arising from the clumps of the protein. Now, it won't only impede the formation of new clumps, it will also dissociate or unpick these. And the way it does it is we tend to think about clumps or aggregates as really static, as once these three are together, there's no pulling them apart. But actually what happens is these three are together and then another big round blob might come along and might join in, and so there's a fourth, but then it might lose one. So then it goes back to three, and then it would add two, and it would go up to five. So they're quite dynamic. They're continuously forming and dissociating and releasing molecules. But the balance is always tipped towards clumping, sadly. So in the end, the structure will normally, in the absence of a tweezer, grow. Now, when the tweezer is actually present, it takes advantage of this dynamic process. And what happens is the minute that this ball is released, it will just stabilize that one. And so it will, in the end, through time, end up dissociating or unpicking or pulling apart these aggregates. Now, molecular tweezers have been shown to reduce pathology in an Alzheimer's disease model, particularly the one that we work with, CLR01. If we look at this Alzheimer's disease animal, we can see that it has a lot of these aggregates or clumps as beta amyloid plaque. This is the brown staining that you can see here, almost showing you the shape of a seahorse or the hippocampus in the brain. So the more brown we see, the worse the animal is doing. In the same way, we can also highlight the professional vacuum cleaners of the brain, microglia, and we can also see that there's quite a lot of brown in this image. Now, this is the case of an untreated animal. When we treat the animals for a month with the tweezer, what we can see is that there's a massive reduction of the deposition of the beta amyloid and the deposition of the aggregates and the clumps. So the cells are much happier because there's less material to fight and to disrupt them. And we can see that microglia have gone back to normal and have gone back to a resting state, and they're not that angry looking anymore. What we have done in the lab is we have expanded this to a different neurodegenerative disease, to Parkinson's, and we have looked at the motor phenotypes and the pathology in the case of Parkinson's. And so if we put animals on a rod and we make this rod turn, at one point it accelerates. At one point it's just too much and the animal kind of can't coordinate anymore and will fall off the rod. In the case of normal animals, this is around 200 seconds. When we look at Parkinsonian animals, they tend to fall off a bit quicker, a bit sooner, because they're not as coordinated as control animals because we've made them Parkinsonian. Now, when we treat these animals with a molecular tweezer, we can restore these effects and we can see them going back to normal levels. If we look at their gait, which is normally impaired in Parkinson's patients, what we can also see is that Parkinsonian animals have impaired gait, and that we can again restore with 2 months worth of molecular treatment these effects that we see in their gait. But perhaps more excitingly, we can highlight the early clumps or aggregates in the brains of these animals with these red little dots or puncta, or control animals will have a lot of puncta, but when we look at Parkinsonian animals, they will have perhaps double as much load of pathology or a load of clumps. And then if we treat these animals with the tweezer, we can see that there's a really drastic reduction in the amount of puncta, which means that in the end, we have a situation, we all get some aggregation because that's just the process of natural aging. In disease, we get too much aggregation, which the brain cannot cope with. And then if we treat those brains with tweezer, we can restore it back to normal levels. Now, hopefully, I've left you with the following conclusions: that molecular tweezers are able to reduce pathology in an Alzheimer's disease animal model, that we're also able to reduce pathology in a Parkinson's context, and that therefore these molecules represent interesting candidates for the treatment of neurodegenerative diseases. And I'd like to thank all the collaborators all over the world that have made this work possible, because it takes a worldwide effort to actually do these studies. And it also takes a lot of money. So thank you to all of the funders that have funded me over the last 5 years, and you for your attention. Thank you very much.